And our ultimate goal with our research is to understand the relationship between the pathogenic variants and the clinical manifestations in KAT6. To start off, I'm going to talk about three main questions that are going to come up throughout the presentation, and that is what are iPSCs, what is an iPSC bank, and how this is going to help us as researchers, patients, and families. So to start off, iPSC stands for induced pluripotent stem cells. Most people are more familiar with embryonic stem cells because they have the ability to differentiate into other types of cells in the body. And iPSCs are very similar, but they are unique in the fact that we are able to isolate cells from a patient and we are able to reprogram them back to an embryonic-like state. And when we do this, we can then differentiate them into different cells in the body. And this seems like a complicated process, but it actually can be achieved over the course of a few months. And so, like I said, we start off by collecting a sample from the patients, and for the Serrano lab, we've been taking blood samples where we can isolate the peripheral blood mononuclear cells, also known as PBMCs, which are a type of blood cell that contain the genetic information. And then we can culture these cells and then introduce a cocktail that contains different factors to reprogram them back to an embryonic-like state. And then we can continue to culture these cells until we have the iPSC cell line. And we put them through a few different quality controls just to ensure that the reprogramming works. And just to show you a quick example, these are two cell lines that we were able to reprogram for the iPSC bank. And this is just one of the quality controls that we do, which is staining for a pluripotency marker. So a big question is, why do we use iPSCs to study rare disorders? Since iPSCs can be derived from a blood or skin sample and then differentiated into different cells or tissues, this allows us to study affected tissues that otherwise may be difficult to access from the patient. And animal research has been very helpful in advancing scientific knowledge, but we do recognize that there is still some differences in the biology between animals and humans. And with iPSCs, we can model human disorders using human cells. And this can also be helpful for multisystemic disorders because we can reprogram and then differentiate the cells into different cell types of the body while maintaining that genetic information so we can study that pathogenic variant in the context of different cells. And so what this looks like for an iPSC bank, like I mentioned before, we can take the blood samples from multiple different patients, isolate the PBMCs, put them through the process of reprogramming to have the iPSC lines. And when we do this for multiple different samples, we can then store them at the iPSC bank. And this is going to be held at the Center for Regenerative Medicine, also known as the CREM at Boston University, which I will talk more about in the next slide. And so our goal with this iPSC bank is to have it be open access, meaning that we're going to share this with scientists and researchers who would like to use these cell lines, which then they can incorporate into their research to look at different cell types and different organoids in hopes of leading to different functional testing in hopes of learning more about KAT6. So like I said, the iPSC bank is going to be held at the Center for Regenerative Medicine at Boston University. And this center is completely open source, meaning every resource that is generated can be shared. And this ranges from the different protocols that we developed to the iPSC lines. And so our hope is that instead of having researchers try and establish these same cell lines, we can help minimize that time by sharing them with everyone so we can get them in their experiments a little bit faster. And you're welcome to take a look at our website and see about the different iPSC lines and resources that we have. So 2 years ago, we started collaborating with the KAT6 Foundation to collect the blood samples. And over the past year, we have started the process of reprogramming. And so this figure here is just summarizing the different samples that we have received for KAT6A and KAT6B, and the samples that we have reprogrammed over the past year. And I just want to take a moment and thank everyone for who has donated thus far and is continuing to donate to this iPSC bank, because it would not be possible without your support. And we are currently doing the blood draw today, so if you're interested, let me know and we can coordinate that. So thus far in the talk, I've talked about how we can collect patient blood samples, reprogram these patient samples to establish the iPSC bank. But now I'm going to switch gears a little bit and talk about how the Serrano lab has been using these iPSCs in our research. So KAT6A and KAT6B are epigenetic regulators, meaning that they can add proteins to histones. And when these proteins or marks are added to the histones, they can change the formation of chromatin. So the chromatin can go from this restricted structure to an open structure. And then this is going to help make the chromatin accessible, which is going to lead to different gene expression and then cellular functions. And this is very important, but can be difficult to understand because there's a lot of other cellular functions that are going on that need to be taken into consideration. And so what our lab is doing is looking to find a way to discover and learn more about what specific marks KAT6A and KAT6B are adding to the chromatin. And so previous techniques and research has tried to look at this, but it can be very difficult because although we can look at what marks are being added to histone 3, there's not much resolution. And what I mean by resolution is that the cell status isn't being taken into consideration. So the cell is doing multiple different things at once. It could be dividing, it could be migrating, it could be under stressful conditions, or it could be under conditions that make it happy. And we want to take that into consideration. So previous techniques can kind of be seen as like seeing a partial rainbow, where you kind of get the structure, where you kind of understand what's going on, but you can't see the specific colors. And so what we've been doing is developing a protocol using spectral flow cytometry, which is a relatively new technology that we have at Boston University, where we're able to see the different histone 3 marks that are being added while getting this resolution, meaning we are able to look at, is the cell migrating, is it dividing, did it just divide, and we're able to take that into consideration. And so we're hoping that this gives us more of the full rainbow, where we're not only able to see the structure, but we're able to see the different colors within it. And so what this means for the bench work and for the experiments is, so we've been taking our KAT6 iPSC lines that we have developed along with control iPSCs, and we're putting them through a differentiation. For the purpose of this talk, some of the information I'm going to show you later is specifically for neural progenitor cells, also known as NPCs, which give rise to neurons. And then we put them through cellular profiling, which is where we're going to be staining for different histone marks and also different activities. And the goal of this cellular profiling is we really want to answer 3 main questions. We want to know if the cells are different. We know we have the KAT6 iPSC lines and the control iPSC lines, and we want to make sure we can distinguish those. Next, we want to know what they are doing. As I mentioned, we want to know if they're dividing, if they just divided, if they're migrating, what are the cells doing in that moment? And lastly, we want to know, what are they? Even though we put them through the NPC differentiation, the differentiations aren't always going to be 100%. So we just want to make sure we're looking at the cell type that we think we are. And so this is a very brief overview of what the protocol looks like. So it starts with collecting the patient samples and putting them through that process of reprogramming and then differentiating them into the desired cell type. And then we can screen for these different marks in cellular processes. And then we put them through the spectral flow cytometry and use a software to analyze this. And with this protocol, we've been able to improve the amount of samples we're able to look at once, and also increase the amount of marks that we are looking at. And so this is just an example of some of the information that we are able to get with this data. So the first thing we do when we have all these cells in the software is we want to ask them 2 questions to start. We want to know what they are doing. And so then the software gives us this map, and based off of the different colors, we can know what the cells are doing. So going back to that cell status, are they dividing? Are they migrating? Did they just divide? And then we're able to stain for specific markers to ensure that they're the type of cells we think they are. So for this example, we can make sure that they are in fact those neural progenitor cells that we thought. And then next, we want to look at the different histone marks. So for this example, we were looking at the histone 3 lysine 9 acetylation. We were looking at the histone 3 lysine 27 acetylation, and we're looking at the histone 3 lysine 14 acetylation. And so this is telling us exactly if these marks are present. But what's really great about this is we can take it even a step further, and we are actually able to quantify these different acetylations. So we can go in and we can look at our control iPSC lines, which are in the gray, and then we have a KAT6A line in the blue, and we can look at how the acetylation levels differ between these different lines. And so this is the point we're at right now, where we're able to quantify these different acetylation marks, and our next step is to be looking at what exactly does this mean. And so this is just to briefly summarize what I mentioned with the spectroflow cytometry, where we're able to take our KAT6 iPSC lines, our control iPSC lines, put them through that cell differentiation, and do the cellular profiling, where we're able to look at, are the cells different? What are they doing? What are they? And we're able to then assess the different acetylation levels. And so going forward, we want to continue to do this, but we don't want to do it in just the neural progenitor cells. Our plan is to do it in multiple different cell types, where we can then ask that same question and look at the different acetylation levels. So I just wanted to wrap up by giving a quick summary of everything that I talked about. In the first half, I talked about collecting the patient blood samples, reprogramming them, and establishing the iPSC bank. And then for the research portion, I talked about the spectroflow cytometry protocol that we developed, how we've been able to assess the histone 3 acetylation levels. And then going forward, we want to look at what does that mean for different types of cells to only have one copy of KAT6A or KAT6B, with then the ultimate goal of understanding how this contributes to the KAT6 clinical manifestations. And I am happy to take any questions that you may have. For the acetylation levels, so we're currently in the process of just quantifying it, and then we're going to look and interpret exactly what does that mean. So we haven't gotten there just yet. So what our goal is to do this profiling in multiple different cell types, and then understand how that variant is specifically working in that cell type. So I can't give like a specific, like, what do I think is going to happen, but we just want to look at that relationship. Uh, I wouldn't say speculation, more of just understanding the relationship. I'm happy to talk more if I'm not answering your question specifically. Yeah. 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 Mhm.
you're finding in terms of Jason, get going, availability in the bank and the samples to the whole research community, very valuable, right? So a lot of this based on infrastructure and the core research is the critical piece. Other questions before we break? I just want to make a statement. I've been involved for a little while. I'm just but I don't add to anything. But from when we started till now, tremendous advances. They may not make sense to most of us because you're all medical, and we may not, but we understand where you're going. And so I say thank you. That's actually a really good segue. Before we break real quick, all the presentations that you've seen so far today are directly supported by the KAT6 Foundation. And so I want to just take a minute to recognize and call out